that we're looking for Neanderthal sequence in. So in fact, there's a lot more of the Denisovan genome to be found. So that's interesting. We can identify intragress sequence. But really what we're interested in is understanding whether admixture was just an interesting side note to human history, or was it something more significant? And in particular, did these sequences that we inherited from Neanderthals and Denisovans, did they have negative fitness consequences? That means, were some of these sequences deleterious? Were some of the sequences advantageous and confer an advantage to our ancestors? And then ultimately, we'd like to know what are the phenotypic consequences of hybridization. And I'm going to focus mainly on these two issues today. So this is an overwhelming slide showing the distribution of Neanderthal sequence that we can find in modern individuals, in European individuals in blue, in East Asian individuals in red. In each place we find Neanderthal sequence in one of these populations, we put a tick mark on the chromosome. The gray regions are just parts of the genome that are too structurally complex to analyze, so we just ignore them. And the black circles are centromeres. And one thing that you might be able to see if you stare at this long enough, and we stare at it for long times, is that there's a non-uniform distribution of surviving Neanderthal lineages. For example, this region, also highlighted by Sriram this morning, is about a 10 megabase region on chromosome 7 that's significantly depleted of Neanderthal sequence. It's also significantly depleted of Denisovan sequence. And what this suggests is that there once probably was Neanderthal and Denisovan sequence in this region, but it was deleterious in modern humans and eliminated by natural selection. And as Sriram pointed out, right in the middle of this region is the gene FOXP2 that's been implicated for speech in speech and language. So if we're interested in the genetic substrates of uniquely modern human phenotypes, these deserts of archaic sequence, I think, are a really good starting point. But not all sequences that we inherited from Neanderthals or Denisovans were deleterious. Some, in fact, were advantageous. And we know that there's somewhere on the order of 50 to 100 places in the genome where there's examples of adaptive introgression. That is, Neanderthal and Denisovan sequences were beneficial and rose to high frequency in the population. And we can find examples of this in all of the populations that we look at. And this is pretty fascinating because as modern humans are dispersing into these new environments, they're admixing and picking up beneficial copies of genes from a species or group of populations that have been there for hundreds of thousands of years before them. And so this is a pretty efficient way to adapt to new environmental conditions. And you can sort of generally say that the phenotypes that were likely influenced by adaptive introgression tend to fall into a couple categories. So things that influence our ability to adapt to new environments, like high altitude, for example. Vast majority of adaptive introgression genes are involved in pathogen defense, and we know that pathogens are one of the strongest selective pressures in humans. And then there's a set of genes that we don't really fully understand that are involved in skin and hair biology, and they too show a very strong signature of adaptive introgression. So we'd like to continue to understand how hybridizing or mating with Neanderthals and Denisovans influence the trajectory of human evolution. But in the last few minutes that I have, I want to tell you about some work that we published just a few weeks ago, actually, in which we developed a new method that reveals a new twist in our understanding of human history in mixing with Neanderthals. And one thing that you might have noticed earlier in my talk is that when I talked about patterns of Neanderthal ancestry, I exclusively focused on non-African populations. So I showed you how much Neanderthal sequence there was in East Asians, South Asians, Europeans, and American populations, but didn't say anything about individuals of African ancestry. And that's because all of the methods up until this point have assumed that Neanderthal ancestry in Africa was either very little or non-existent. And so we recently developed a new method that didn't make this assumption. And so we were excited to apply it to individuals of African ancestry. And to our surprise, we actually found substantial amounts of Neanderthal sequence in African individuals. And these were the 5 populations that were available for analysis from the 1,000 Genomes project. Purple here represents African admixed individuals, so largely African Americans. But even in these African populations from the 1,000 Genomes project, we find about 17 megabases of Neanderthal sequence per individual. And just as a comparison, when we look at sort of the same individuals and call Neanderthal sequence using previous methods that we developed that make this assumption that there's little Neanderthal ancestry in Africa, we only call maybe 500 kilobases, so like 2 orders of magnitude less. So this was a really strong signal, and it was very surprising. So we do see Neanderthal ancestry using this new method, but what explains this signal? Well, to make a long story short, there's really 2 primary explanations. So the first is that there were migrations back to Africa. So people left Africa in the major out of Africa dispersal, hybridized or admixed with Neanderthals, and some returned back to Africa carrying the Neanderthal sequence with them. And our results show that the amount of back migration has probably been much larger than we've previously thought. So that's one part of the signal. The second part, actually, is really fascinating, and it's something that we really wasn't on our radar until we got this result. And that is that part of the signal of Neanderthal ancestry in Africa is due to an early out-of-Africa dispersal and gene flow from humans into Neanderthals. And so let me unpack that a little bit for you. So this is a simple phylogeny showing the relationship between Neanderthals and 3 modern human populations, so Africans, Europeans, and East Asians. And so the bottom here represents the present, and we go into the past as we go towards the top. And this hatch mark just is to indicate that the times aren't going to be drawn proportionally. So we know that Neanderthals and modern humans split around 600,000 years ago. And what our data shows is that not only was there this out-of-Africa dispersal that happened 80,000 years ago that resulted in the peopling of the world, but there was also a much earlier dispersal of humans out of Africa, around 200,000 years ago, and they encountered Neanderthals and admixed with them. So in fact, some of the sequence that we call as Neanderthal, it's not Neanderthal sequence in modern humans, it's that Neanderthals have modern human sequence. And so this adds a further twist to sort of this complex pattern of admixture and gene flow and arrows pointing in every direction. So in conclusion, there is substantial amounts of the Neanderthal and Denisovan genome that remain in modern individuals. There were fitness consequences to hybridization, both good and bad. Humans, Neanderthals, and Denisovans have mixed multiple times, likely in multiple places, and that there were multiple dispersals both in and out of Africa. And I think this last point is something that is really important in genetics, is that we often have simple models of how humans dispersed around the world, and that the more data we look at, the more complex these models become, and that it's important to take into account the dispersals both out of Africa and back into Africa to really understand patterns of Neanderthal ancestry. So I would like to acknowledge my lab, in particular Ben Verneau, who did a lot of the early work on finding Neanderthal sequence, my collaborators, and my 2 boys, who I'm sure are not watching right now. Thank you very much.